In progress.
So now these are all quantitative traits, polygenic traits. Now Mendelian traits. Mendelian traits are qualitative traits. Qualitative traits. So differentiation only, just differentiation. So you can see here single loci or single gene with two alleles. Or else you don't need to write single loci or single gene controlled traits. We call as a Mendelian traits with law of dominance and recessive. So law of dominance and recessive. So these locis, they must follow law of dominance and recessive. So we had seen so many locis are there. So HBA, HBS. So HBA and HBS both are single loci, but they are incomplete dominant. So all the locis which follow law of dominant and recessive, those locis we call as a Mendelian locis. So inherited as a discontinued traits. So discontinued traits. So discontinued traits means either or comes, not in between. So intermediary things never appear here. So either that or this, but not the intermediate thing, it never appear. So a person, a child, so a child born to a couple, so taster and non-taster. Okay, taster and non-taster. So these are all the couples. The child must be either taster or non-taster. Intermediate, it never possible. Understand? So that's why we call it as a discontinued traits. So here, so additive effect, equal contribution, but here there is no additive effect. So no additive effect. Quality. So no additive effect. So the gene quality expressed or not. So expressed or not. So that only we will observe. So sometime expressing additive effect, these kind of things are not present. So these we call as a qualitative traits. Qualitative. So we cannot measure, but we can observe the whether the property is exhibited or not, but we cannot measure that. So qualitative traits. So environmental role of environment is absent. So role of environment is absent. So how can we say role of environment is absent? So you can take the blood groups. So blood groups generally formed during the fetus time. So fetus. So blood groups generally formed during the fetus time. But so fetus time blood groups come into being. Even after birth also. That means neonatal time, okay, infancy, childhood, okay, whatever the stage, whatever the environment, even diseases, environment, any kind of things never bring change in the blood group. If a person during the fetus time, that A or B group or O blood group, any kind of blood group they acquired. So it is lifelong till his last birth. So the same blood group is found. It never subject to change. But skin color, if a person residing in a equatorial climate, by birth, if he acquire dark skin color, by moving after the birth into polar region or temperate region, so there is a slightly change in the skin color we can observe. That means when environment change, there is a change in this quantitative trait. But whatever the environment, whatever the nutritional condition, disease effect, so none of the factors bring the change in the traits. That we call as that means there is no external environment role. A person is a taster during the fetus time. Lifelong he is a taster. So sometimes he is a taster, sometimes non-taster, again coming a taster. So these kind of things are never present. Okay, that means the role of environment here. So it is forever. Role of environment is absent. It cannot bring any change once the character exhibited by the gene. So examples we can quote as taster of PTC substance, roller of tongue, hairy ears. So hairy ears. There are so many. Diabetes. So diabetes. There are so many examples we can give as a Mendelian traits. So diabetes, can it impacted by the environment? So we cannot observe any plasticity. No plasticity. Non-plasticity genes. So non-plasticity gene. So when compared to polygenic gene, so these genes, though it have some adaptive value, but adaptive. So it do have adaptive value. So you know about wild gene. So wild gene means what? When it comes to ABO blood group. So O is the wild gene. So O is the wild gene through which sometime A allele come into being, sometime B allele come into being. Now overall human population has A allele, B allele, O allele. So without having any advantage, nature never promote these kind of new alleles. Okay, so here, so polymorphs are present. Okay, here polymorphs are present. So what do you mean by polymorphs? So human beings are polymorphs due to mutations in Mendelian locus. So this is all about polymorphs versus polygenes. If somebody asks, what do you write? Polymorphs versus polygenes. Polymorphs versus polygene. So polymorphs means, see, chromosomal 9th pair, one of the loci is responsible for blood groups formation or the antigen present on the RBC cell surface. Okay, so human beings are classified into, see, A blood group, B blood group, AB blood group, O blood group. Understand? But some of the individual, so this loci, some of the individual as A, in some of the individual this loci is B, some of the individual AB, some of the individual O. That means the same loci which has four phenotypes due to, so we can say multiple alleles on the loci. So A allele, B allele, O allele. So this we call as a polymorphism. If same loci due to multiple alleles, if here only one allele is there, that is O allele, all human beings are monomorphs. Due to mutations in the wild gene O, A and B alleles come into being. So these are recombination occurred on the loci. Human population classified into multiple morphs or multiple forms with respect to single loci which give the blood groups. So polygene means all the genes give single character. All the loci give single character. So polymorphs means it is the Mendelian loci. Due to mutation, it has multiple alleles. These multiple alleles result into the human beings are classified into multiple forms. So this is, again, they might confuse you, like by giving the polymorphs and polygene, they might confuse you. Polymorphs means same human beings are, if Mali struck, is it strucking again? So, Praniti, are you getting any problem?
No, sir.
Hello? Okay. So, Vijay, it might be internet issue from your end. So, she is getting fine. Okay. So, that means they might twist you like, twist you like. So, use mobile data. So, qualitative traits versus quantitative. Qualitative versus quantitative. Okay. So, second thing is polymorphs versus monomorph. Polymorphs versus polygenes. So, Mendelian genes versus, okay, polygenes. And fourth one, so you can see continuous, discontinuous versus continuous. So they might use different terminology, but the answer is same. Understand? The answer for all these questions are same. So to confuse the students, they use the different terminology. So you should be careful about this one. So this is all about the second one. So another exception in the Mendelian laws was genetic imprinting. Another exception is nothing but genetic imprinting. So what do you mean by genetic imprinting? So genetic imprinting. For suppose, if you take the color blindness or hemophilia, so hemophilia or any kind of genetic disorder. So if couples got married, okay, father passed this gene to the sons, daughters. Okay, so father to daughter, father to son. So two son and daughters are, so they have no genetic disorder. Okay, one son and daughter has genetic disorder. So now the same effects, the same effect, the same symptoms. So the boy, that means son, experienced the same symptoms, same time period, same severity of the daughter. That means normal genetic disorders never show any differentiation between the genders while they are expressing. Understand? So these are all Mendelian traits, Mendelian disorders. Okay, Mendelian loci malfunctioning result into the some kind of disorders. So the same, if the diseases come from the mother, mother to son, mother to daughter, the same effect. Whether the source of inheritance of genetic disorder is father or mother, same severity, same time, same symptoms expressed. So that if same symptoms are observed in the daughter, it is coming from the father. Same symptoms observed again in the another daughter, if it is coming in the mother, can we qualify to call it as a different diseases or same diseases? So daughters are inherited the disease from the father or mother. If both daughters are experiencing same symptoms, the disease name is the same, severity same, time period same, symptoms same. We don't call these are all two different diseases, same diseases. Understand? So in Mendelian, so if your trait is passed from father to daughter, okay, daughter to son, okay, again son, again son. So again son to daughter, again son. So there is nothing like, so whatever the gene effect here, effect, severity, time, the same will be observed here. The same will be observed here. The same will be observed here. So across the generation, these Mendelian disorders have same in nature. That's why the same disease is passed across the generation. We don't name it as a separate names in every generation because symptoms are same in every generation. It is reappearing. That's why hemophilia is appeared in every generation. So this is how we call it. Understand? So now see, these kind of genes are like 2 to 3,000 genes. Some genes are there in our entire genome out of 22,000 genes or 23,000 genes. Different books they call different number. So out of 23,000 genes, so 2 to 3K genes are Mendelian genes. Mendelian genes. But 200 to 300 genes are there. They are imprinted genes. So imprinted genes. So what do you mean by imprinted genes? So they are not Mendelian genes, imprinted genes. For suppose this is the father. So male person. Okay. So he got married a female. So they participated in their reproductive activity. So during that time, so that father sperms produced. So this sperms produced is nothing but a meiosis. So meiosis means DNA replication. So DNA replication through which 2 cells come into being. 2 cells again undergo meiosis to 4 daughter cells come into being. So those daughter cells we call as a sperms. And in this, one of the chromosome has the genetic disorder. So genetic disorder. Okay, mother ova is normal. Mother has no genetic disorder. She released ova. Both are fertilized. Both are fertilized. And the person is female. Female. That means the source of, see, obviously the disease inherited to the daughter and the daughter experience the symptoms. Daughter experience the symptoms. Okay. So for suppose 30 years, she started experiencing the symptoms and the symptoms getting severe. And by the time of 35 years, she died due to disorder. So this is the case. But before expressing the symptoms, she got married and she produced a child. So that means she married another person, male. So now mother has the disorder. So from mother to child, child acquired. Understand? So now the trait is passed to the son. So now son expresses these disorder at 40 years, 40 plus years, less severity. So less severity. Okay. So that means he survived up to 60 years. He survived up to 60 years without dying. So it means when the source of gene is from the father, that is male to female, it is early in appearance. Symptoms are very severe. Within 5 years, he died. Okay. So now when the source, same gene, that gene now mother to son. When mother to son, so when should the gene is inherited to the here? When should it appear actually? Same 30 years it should be appeared. By the severely it will be affected, and by the time of 5 years that son has to be died. But here it delayed. So it delayed, less severe, so that lifespan increased. So that means what happened? So while the gene transmitted from father to daughter, okay, so here this mechanism we call as a genetic imprinting. So how it is imprinting means the gene that mother, this daughter received from the father, she has to produce ova. Ova means meiosis. Meiosis means it's kind of DNA replication. During the DNA replication, whatever the gene received from the father, she erases it. And she imprint her own copy in that gene. Understand? So that means each gender, while they passing their gene to the next offspring, whatever the gene they received from their parents, they erase it. So now if this person passed to the next generation, so the same gene traveled here. While he is producing the sperms, he erased the characteristics received from the mother and freshly he applied the imprinted on the same gene. It is passed to the next generation. So now again, symptoms are very quickly severe. So this is how. That means each gender, while they are transmitting some of the genes to the next generation, whatever the genes they received from their parents, they erase it and freshly imprint their own copies. Such kind of genes we call as a genetic imprinting. Such kind of genes are genetic imprinting. So Mendelian genes never be erased and never be separately imprinted. How many generations that try to pass, the effect is same. But some genes in each generation, it's look like a freshly existed behavior, freshly happening the things. How it is means DNA methylation. So that means in every person, while they are undergoing the DNA replication, so DNA replication. So DNA replication means it is the one copy of gene creating its own duplicated copy that we call as a DNA replication. The precursor of this DNA replication is nothing but DNA methylation. So DNA methylation, it is one of the enzyme. So DNA methylation is a one of enzyme. When it produced, the DNA replication enzyme starts to produce. When this DNA replication touched this DNA and it created duplicated copy. So now you can see the DNA methylation enzyme quantity in oogenesis or ova formation time is more. So in human being, male we called as a spermatogenesis. Female we called as a oogenesis. Oogenesis. But these two are meiosis only. So in female DNA methylation, the enzyme quantity is more. So the same DNA methylation occurred in the male, but the total quantity is less. When more methylation is there, this methyled enzyme is applied on the genes. So those genes which received from the parental, they arise due to or they mask due to heavy methylation. Understand? So in male, for suppose father DNA methylation is less. When and it is passed to the daughter. Okay, so then because of due to less DNA methylation by the father, the disease started early in the expression. But while she is forming her own ova, she prepare her own DNA methylation, the same genes are heavily methylated. So these heavily methylated genes are again passed to the son. So whenever heavy methylated occur, expression is delayed. Okay, expression is delayed. That's why this person delay in the expression of the symptoms. But again, he is a male. He has to produce the sperm. He will apply less DNA methylation. And again, the symptoms are quick. And she produces her own DNA methylation. And she gives the more DNA methylated genes in the next generation. The symptoms are late. Understand? So that means DNA methylation is the reason for the initiation of the meiosis and DNA replication, but the quantity produced in the male and female are differ. More quantity of the DNA methylation, less these genes are expressed, are delayed in the expression, or less DNA methylation, these genes are quickly in expression. Now, what are all these genes? So these genes are gender specific genes. So gender specific genes. So though these genes are present in the male and female, but their behavior change based on the source of that gene inherited by an individual. If this generally, each gender differently stamping genes are received from the mother, so their expression is late. If gender stamping genes are received from the father, their expression is very early. Understand? So these kind of genes are how many present in an individual? 200 to 300 genes. Understand? So now we can take the example. So what do you mean by genetic imprinting means? Genetic imprinting means, so while passing their genes, father stamps differently, mother stamps differently, and these genes are inherited by a child. If mother genes are dominating, they expressed in the late. If father genes are dominating, they expressed in the early. And while these genes are transmitted to the next generation, so they are late in early. So that means any disease causing gene inherited from father to daughter or son. So their symptom, the source is father. Their symptoms are early, severe. So if the source is again from daughter to son or grandson, their symptoms are late. Again, son to son or son to daughter, their symptoms are early. Why? Because the source is male. Here the source is female. Understand? So the same gene present here, okay, present here, but here early expression, here delayed expression. Here severe, here less severe. Here quick death, here late death. So symptoms also vary. Here one type of symptoms, here another type of symptoms. So now we can call it as a two different of diseases. Understand? Two different of diseases. That example I am going to give you. So Huntington chorea. Huntington chorea. Huntington chorea, if it is passed from father to offsprings, such as sons and daughters. So this Huntington chorea disease inherited by both sons and daughters, both are quickly expressed with severe symptoms and death, quick death. Okay, the same gene, if it is inherited from mother to offsprings, son and daughters. So sons and daughters, because it is a autosomal dominant, any gender can be inherited, son and daughter. So delayed in the expression, mild symptoms. So delayed in expression, mild symptoms. So delayed in the death. So delayed in death. So that means the gene is same. Why? Because while he is preparing his own sperms to giving to the offsprings, he will apply less methylated. So less methylation. So less methylation. So here mother applies more DNA methylation. More DNA methylation. So same gene, but expression is differ time wise, symptom wise, severity wise. So this is one of the gene we can say genetically imprinted. So father imprinted one manner, mother imprinted one manner. Understand? So if you take another thing, okay. So Prader-Willi syndrome is there. So father, one of the deletion disorder we call, one of the deletion, if any one of the pair, it has some deletions chromosomal during the crossing over time, some portion is deleted. If the deleted portion present in the father, so that disease is called as a Prader-Willi syndrome. So Prader-Willi syndrome. So Prader-Willi syndrome. If the same disease will be inherited from the mother side, so that we called as a Angelman syndrome. So Angelman syndrome. I will explain. Wait. So Angelman syndrome, mother to offsprings. So Angelman syndrome. So you can see how it is different. So different names means different symptoms. So in Prader-Willi syndrome, hypotonia, so obesity. So third one you can see hypogonadism. So these are all the basic characters of the Prader-Willi syndrome. So Prader-Willi, W. So next one is, so Angelman syndrome, epilepsy. So epilepsy, tremors. So smiling facial expression. So smiling facial expression. So what do you mean by this? I'm going to explain. So father has sixth pair of the chromosome. Okay. So mother has sixth pair of the chromosome. So father has the sixth pair of the chromosome, mother has the sixth pair of the chromosome. I am saying, like, for suppose, imagine. So here some of the portion deleted. So because it is the father, so the deleted chromosome is inherited by the children. So that symptoms are Prader-Willi syndromes. Understand? So if mother is healthy, mother has no deletions, father has the deletion, for say in the second chromosome of the sixth pair, second loci, there is a small deletion. So that result into Prader-Willi syndrome because the deleted chromosome or shortened chromosome received from the father. So what kind of symptoms it expressed means hypotonia, obesity, hypogonadism. So now in second case, father is healthy. Okay, father is completely healthy. There is no problem. So father is absolutely healthy. So sixth pair, father. So in mother, sixth pair, second chromosome, a deletion occurred at the second loci. So now in this case, the children born under the defected or shortened or deleted portion of the chromosome inherited from the mother. So now the mother inherited chromosome or deleted fragmented chromosome, it expressed the epilepsy, tremors, smiling facial expression. So this they named as a Angelman syndrome. So now you can see the deleted portion is same on the sixth pair if it is from the father side, on the sixth pair it is from the mother side. The same loci for the both parents, but it should be shown the same kind of symptoms. When the source of father is Prader-Willi syndrome, when the source is the mother, the syndrome is called Angelman syndrome. Why? Because during their DNA methylation, the gene character changes. Understand? So this is how. So by saying that, okay, if a person is experiencing, for suppose, so these both are autosomal disorders. Autosomal disorders. Okay, so both are autosomal disorders. For suppose there is a disease called, so what we can take color blindness or else. So any disease you can name it, which we were discussed as part of. So any disease, okay, it is coming on the offsprings. In Mendelian, we cannot identify the source of the parents. Autosomal dominant disease generally coming from the father side or mother side, and the child is suffering with the autosomal dominant disease, and we cannot say who is the source. Why? Because it can be possible from inheriting the father side and mother side. So at the same time, so these are also autosomal dominant disorders. Okay, in one disorder symptoms are varied, in another disorder symptoms are varied. So by going these symptoms, if a person is experiencing like epilepsy, tremors, smiling facial expression, confirmly we can say the deleted chromosome received from the mother. Okay, if a person has hypotonia, obesity, hypogonadism, we can say that the symptoms are generally received from the father. Understand? So remaining autosomal dominant and recessive disorders, we cannot pinpointly saying that who is the responsible, either it is a father or mother. But here in genetic imprinted genes, if Huntington chorea showing its symptoms at 20s and 30s, that is the father, from father it is inherited. If Huntington chorea showing symptoms at 40s or 45, the source of inheritance is from the mother. I think it is blinking. I don't know exactly. It is blinking. So now we can see. So genetic imprinting means, so it is the each gender, while they are transferring their genes to the offsprings, they stamp according to their gender. So that we call as a DNA methylation. In female, the DNA methylation is more, the imprinting is more. In male, DNA methylation is less, imprinting is less. So these kind of genes are how many? 200 to 300 genes are there overall in 23,000, but these are all non-Mendelian. So non-Mendelian traits. Why non-Mendelian traits? If the source of origin is father, Prader-Willi. If the source of mother is also same the source is mother, then the mother also have the same disease like Prader-Willi. We can say it is a Mendelian trait, because she stamped differently. That's why symptoms are different. So now it is visible or not? Now it is visible or not? I'm not sure why it is getting. I have to do research. It should not get generally. Okay, so this is genetic imprinting. So generally, not all the genes are imprinted, only few genes are imprinted. These imprinted genes are inherited from the father, one type of expression. If these are imprinted from the mother, another type of expression. Same loci, based on the gender, its effects vary. That we call as a genetic imprinted genes. So why these genetic imprinted genes? So now it is coming or not clearly? So whether it is clearly coming or not?
Yes, sir.
Okay. So now we can say that if a gene is from mother, delayed expression. Okay. So slow symptoms or light symptoms. Light symptoms. If the gene is from father, early in expression, okay, severe in expression. So this is the means the source of, based on the source, if a disease has severity and quickly appeared, the source is father. If a severity is slight as well as delayed expression, the source is mother. So any kind of, if these kind of diseases can be identified, they were caused by differential imprinting. If it is from mother or father, they have both severity, same time of expression. Those diseases we call as a Mendelian diseases. We cannot pinpointly say who are the source of origin. So now the theories. Now the theories are general understanding. Why these imprinting necessitated or it is required? Why certain genes are imprinted? That is also hardly 2 to 300 genes, not like all the genes. So why? So that means, for suppose, so growth promoting genes are there. So growth promoting genes are there. So the theory name is like parental conflict. Parental conflict theory. So this only you can remember. You know, generally nobody discuss all these things. You should write this only theory. Parental conflict theory. For suppose, growth promoting genes are gender based imprinted. Gender based imprinted. That means here, in one of, see, if one of the gene, it has more quickly growth promoting. So this is the low key. Father gene is present, mother gene is present. If father gene, the father gene coming from the father gender, less methylated. So methylated, and mother gene coming from the mother, more methylated. So more methylated. So now you can see this gene, this loci is related to growth. So this gene is related to the growth. Understand? So if mother gene could not able to do any impact, then what would happen? So this gene promote the fetus inside the womb, promote the fetus, use more resources of the mother body and grow in size. That means quickly expressed. That means it is taking more nutrition, quickly try to gain the more weight, so that more harvestation of the resources of the mother. If that could happen, the mother immediately getting weakened. So it should be countered. For that purpose, mother applied more DNA methylation. This gene is countered. Just wait. This womb is not only for you. There are upcoming babies also there in the future. You can only use less resources by limiting your growth. So this is how, if there is no counter, all the father genes are quickly in expression, all resource harvestation. Mother gene is countered. You could only utilize how much it is required, not all the resources, because it is also for future generation. So these kind of parental conflict, because both parents are different genders, genes are from different different genders. So each gene, if is trying to do overactivity, another gene is trying to balance that one. So this is how gender based differentiation of the or imprinting of the genes are necessitated. So this is one of the parental conflict theory you can explain. Understand? So these are all species specific. In one of the species, if growth genes are imprinted by the genders, in another species, growth genes may not be imprinted. Okay, so it is generally appeared on the mammalian species are more. Mammalian means placenta, nutrition taken from the mother. Mammalians, so that is mammary glands are there. So these species have gender based imprinting genes. In another species, the scientists never observed any kind of imprinting genes. Only mammalian species have. Okay, the same genes never be imprinted in the different species. So different species have differently imprinted genes based on their resource utilization. Okay, it is mammalians are more in observed, and these are all erasable. So erasable means if a gene is received from father, mother to son. So during son formation of his own gametes, okay, he will erase the whatever the copy coming from the mother. He will apply his own imprinting. Okay, this gene is passed to daughter. So daughter, while forming her own ovas, okay, she will apply her own imprinting. That means in every generation, this imprinting has to be erased. So this we call as genetic imprinting. So this we call as a genetic imprinting. So by this topic, we finished the Mendelian genetics. So if you have any doubts, you can let me know. So I will find it what is the exact reason. Okay, generally it is like plug and play. So I will switch on and take it, but sometime I don't know why it is coming. I have to do some research. It is software side and internet side or any other reason. Okay, so tomorrow is there any class? or not, I will let you know by morning time. Okay. So if you have any doubts, you can text me on WhatsApp also. I will respond. I will explain the next class also. So till now, if you have any doubts, you can raise it. So till now, so we did like what are all the exceptions of Mendelian genetics? Exceptions of the Mendelian genetics are, I am recollecting one more time. So gene interactions. So gene interactions are we called as a epistatic genes. Epistatic genes. So sex linked, sex influenced, sex limited, sex limited. So polygenes. So polygenes. Next, imprinted genes or genetic imprinting, imprinted genes. So imprinted genes or pleiotropism. So pleiotropism, single gene, multiple outputs. Single gene, multiple outputs. So they might ask each one for 10 marker, or they might ask like all together for 20 marker. So genetic imprinting, one of the year it came for 15 marks. Polygenes, it came for 20 marker, one of the year. So pleiotropism, gene interaction, they came for 10 marker. So each one is worthy of independent question. At the same time, they might ask like discuss various gene behaviors which are all not following the Mendelian properties. So sex linked are X dominant and recessive, Y inheritance. Sex influenced are baldness. Okay, sex limited are milk secretion, only female has. So polygenes, skin color we observed, quantitative. Imprinted genes we have seen Huntington chorea, if it is coming from the father early, it is coming from the mother late in the expression. So at the same time, Prader-Willi syndrome, if it is coming from the father, or Angelman syndrome, if it is coming from the mother, symptoms are different. So these are all the example. The reason is like DNA methylation. So mechanism is like genetic imprinting mechanism is based on the how much quantity of the DNA methylation they produce, or genes are applied DNA methylation. Okay, genes are applied DNA methylation. So reasons are possible explanation. Paternal and maternal genes conflicting in their expression. It might be the source. They have adapted by one is growth promoting, another one is growth limiting. So that is the source in the DNA methylation. Understand? So this is all about Mendelian genetics. So if you have any doubts, you can ask or you can text me. In next class, we will discuss. So this issue, I will find it out.